Hey everyone, my goal for this video is to clarify what the word seed means. It is a word that is thrown around a lot just by common folks, and the word seed is used in other ways, but we're talking about what is a true botanical seed today. And I'll tell you, it's definitely not a spore of any kind. There's a spore involved, but we'll get to that. The complexity of seeds is taken for granted, right? Like we eat seeds and it's like, wow, cool. It's just regular food. No, it's an amazing morphological structure that has evolved to protect the young of the next generation. And that's what evolution is all about, is getting your genes into the next generation. So here are our learning goals for the day. We need to understand how a seed is 3 different life stages in one structure. Yeah, wow. It's 3 different life stages in one structure, by the way. Okay, and how the relationship between, we need to know the relationship between an ovule and a seed. We need to learn how seeds develop and how pollen grains develop. They're kind of connected. And then we need to understand the relationship between megaspores and cellulonella, which we learned about last week, and the seeds of gymnosperms. So here's your vocabulary. Some new stuff, but not very much, actually. And here's the contents of what we're going to be talking about on this video, sort of in this order. First, we're going to talk about the evolution of the Megasporangium, and then ovule anatomy, and then the evolution of the microspore, and then pollen anatomy, which, spoiler alert, a microspore is a pollen grain. And then we're going to talk about seed anatomy, and we're going to bring it all together, sort of summarize it all using the gymnosperm life cycle. Okay, so let's recap from last week what the strobolus of cellulonella looks like. Now, to be really clear, cellulonella does not have a common ancestor with gymnosperms that had heterospore. This was the cellulonella version of heterospore evolved sort of independently, but we probably don't know exactly how the ancestors of gymnosperms version of heterospore evolved, and so we can use cellulonella as sort of a proxy to think about maybe how that happened. And then there's also some fossil evidence for some of the things I'll be talking about. So remember, in cellulonella, we had these 2 different types of spores, and we had these microspores up here, and then we had these sort of larger megaspores. And remember that in cellulonella, these megaspores, they're just released from the Megasporangium. So maybe in this case, on this picture, the Megasporangium is this whole kind of structure right here. And it is sort of subtended by one of these pointy things. Remember, this is one of those sporophylls, a leaf that's associated with the sporangium. Namely, that's a megasporophyll. And this megasporangium will sort of open up and just release the megaspores. Oops, again. So there will be sort of a little opening in the megasporangium, and then these megaspores would be released. And then remember, inside of the megaspore, we have haploid cells that are going to undergo mitosis to form a gametophyte, right? Remember that? And then the gametophyte is sort of this free-living structure. Yeah, we saw that in cellulonella and a few other things. Okay, so again, that megaspore, it lands on the ground somewhere. And right here it is sort of floating through the air. And then a megagametophyte sort of germinates from inside of the megaspore. And this is all haploid. The megagametophyte is what will then produce the archegonia. So this is all happening somewhere in the environment, right? This megagametophyte is sort of on its own besides the megaspore wall. And so it's going to need to be dealing with hydration on its own, and who knows what other environmental conditions it'll need to deal with on its own. And right, by on its own, I just mean sort of like it's just like this naked gametophyte, more or less. But the one thing that is protected, or that was protected in cellulonella, still is the egg, right? The egg, like we have seen since all of the land plants, the egg is retained, and therefore the zygote is retained in the megagametophyte. Cool. And then, then the sporophyte just sort of had to grow right out of that megagametophyte that was somewhere in the environment. So the sporophyte is limited to growing where that gametophyte was able to grow. Moving on. Now, we're really going to talk about the nitty-gritty of the details of the evolution of a megasporangium, sort of over millions and millions of years, and so many countless generations of these organisms. They ended up having sort of selective pressures for the things we're going to be talking about right now. So in some hypothetical evolutionary ancestor of the gymnosperms, just similar to what Selaginella had, the Megasporangium had 4 megaspores in the Megasporangium, and it looked something like this. Well, where do megaspores come from? Well, besides the fact that they come from the Megasporangium, they come from a single cell called a megasporocyte. So at one point, there was sort of this one cell, and it became these 4 megaspores. Okay? Well, again, over evolutionary time, there was more sort of reduction. And what happened was there was just a single megaspore per megasporangium. Now, you're thinking, well, megaspores are the result of meiosis, right? It was this, going back, it was this megasporocyte, that one that I had drawn up here, I forgot to label it. It was that megasporocyte that underwent meiosis to form those 4 megaspores. And so what happens is over evolutionary time, yes, we still have 4 results of meiosis, but 3 of them just sort of abort, and they just sort of, one of them just survives, and then the other 3 just sort of go away. And then another thing happens, evolutionarily speaking. Again, this is not happening in a single generation. There's evolutionary pressure, selective pressure for this. Maybe it just sort of made sense to put all of your eggs in one basket, if you will, and have just this one functional megaspore. Well, then the next thing that happened was the sporangium was no longer opening. It no longer opened to release the megaspore. What? So yeah, notice between these 2 slides, this one was, as you would call, dehiscent, this evolutionary ancestor, had a dehiscent megasporangium because it opened. This one is indehiscent because it does not open. And now that one big functional megaspore is enclosed. It's sort of stuck inside of that megasporangium. And well, that's actually a really convenient place for it to be, turns out. So again, over evolutionary time, many generations, sort of there's this other layer of tissue that forms around the outside of that megasporangium. And this is called integument. The integument is what's labeled in green here on the very outside here, and it's a full layer of protective tissue around that megasporangium. Now, since we know that eventually, at some point, this is, the megasporangium is what's going to form the megagametophyte and therefore an egg, and we're going to need fertilization to take place at some point. And so we're going to need some sort of opening. And so there is this little opening in the integument, and it's called the micropyle right there. So this is what an ovule is. An ovule is an indehiscent, integumented megasporangium with one functional megaspore. Okay? And we are going to expand on this. First of all, how do we even know? How do we know that this evolutionary story that I just told you is even true? Well, we actually do have a plant fossil record from about 350 million years, and this is a scientific artist's rendition of a fossil that was found that is about 365 million years old. And these might have been the sort of some of the earliest ovules. And there are these lobes of integument, these protective layers, like I said, and they sort of fused over time. And this is actually, we have multiple lines of evidence of these different extinct fossil taxa that people have studied and realized that actually, yeah, we do have a pretty complete story of pretty much exactly how an ovule evolved over time. Now, this is very convenient because we don't have any of the living descendants of any of these fossil recorded early ovules. So we're talking about something that happened, 305 million years ago or so. This is back when the lycopodiophytes were sort of dominating the landscape. Okay, so back to this. Why is this important? Why is an ovule even important? Well, an ovule is what's going to become a seed. So this whole talk is about a seed, right? Well, once an ovule has undergone fertilization, which I know we haven't even talked about the egg yet, we're going to get there, that's what a seed is. Right? A seed is a fertilized ovule. Fantastic. So now let's redraw this realistically so that it looks like a more realistic version of a extant gymnosperm. So switching to ovule anatomy now. The dark black here is the megasporangium. Remember the megasporangium? It just never opened up. And so that megaspore was retained inside. And now this is all happening on the female strobolus, by the way. This is all happening on a strobolus. More on that later, but right now we're really focusing on the ovule. Okay, so what's inside of that megaspore, you ask? Well, a megagametophyte. Just like the megaspores of, all the other things we've talked about so far. The only thing we've talked about so far, Selaginella. But the regular spores of homosporous plants like the ferns and horsetails. Those homosporous plants, the spores would produce a gametophyte. Here, a megaspore produces a megagametophyte. It's the same story, except for this time the megagametophyte doesn't need to be on its own in the environment. No, it's sort of retained and nourished within the megasporangium. So the other word for the megasporangium is the nucellus. So I'm going to use those words interchangeably. And, since we're talking about, the megasporangium, this is the structure that was housing, or that is housing that megaspore. The megaspore is the result of meiosis. And so the megasporangium is sort of in the previous life stage. In other words, the diploid life stage. Right? The megasporangium here is diploid, 2N. But the megaspore itself is N, haploid, which makes sense because it's going to go through mitosis to form a gametophyte. So let's talk about that now. The megagametophyte is endosporic. Again, that just means that it never emerges. It's formed inside of something, and in this case, inside of the megaspore wall membrane. So then what's inside of the megagametophyte, you ask? Well, just like on all the other gametophytes we've talked about, archegonia. Yeah. An archegonia, and then what's inside an archegonium? An egg. So no surprises there. It's just the surprising thing is that this is all happening not only inside of an enclosed megasporangium, but attached to the strobolus of the sort of the mother sporophyte plant. Okay. Got a little sidetracked there. So back to this diagram. The archegonium is what is going to produce the egg, as always. So here the archegonium is just sort of indicated by this sort of outline of cells, and then the egg is that that structure that's actually going to undergo fertilization at some point. That one cell that's going to undergo fertilization with the sperm cell, if it's lucky. Okay, so we've got lots of layers here. Make sure you kind of can remember which ones here are diploid, which ones are haploid. Remember everything that's sort of, we just talked about it. So see if you can draw this out and then label these from this very figure as haploid or diploid in your notes. So this is a mature ovule. Fantastic. This means it's ready for fertilization. And so once an egg undergoes fertilization, it's going to form a zygote. That's no news. And then the zygote's going to undergo mitosis, which forms an embryo. There's nothing new here. The embryo's going to undergo mitosis and form a sporophyte. So what is new here is that we've got these terms ovule and seed. An ovule is the sort of structure that houses the egg. right? And then the seed is sort of the structure that houses the embryo. But the seed includes sort of the embryo. Okay. And what's really cool is that megagametophyte stage is retained sort of in between these two diploid stages. Okay. So what does this really mean? How does this happen? How can we bring this a little bit more together with the other side of the story here, the male side of the story? Well, we know that sperm cells need to get to these eggs somehow. And so how will they do this? This is going to require a new process called pollination. Pollination is sort of the act of pollen grain sperm cells really moving from one organism to another and not swimming there. Maybe there's possibly some flagella involved, but in certain gymnosperms, but they are still enclosed in pollen grains. Pollination, which uses pollen. What is pollen? Pollen is just another name for the microspores of seed plants. Okay, so this is some evolutionary ancestor, some hypothetical progymnosperm microspore. Okay, so in this hypothetical ancestor, we have this microspore wall. Inside of it, everything is, of course, going to be haploid. We have these things called prothallial cells. We'll get to those in a sec, but just know that they were sort of there in the ancestor. We've got this antheridium, and this antheridial jacket layer. This is, remember, this sort of the sterile layer of cells that surrounds the gamete-producing structures. Well, that was present in the ancestor here, and then of course there were lots of sperm cells, and together everything that's in yellow here. This was the microgametophyte. Well, a living seed plant microspore, which we call them pollen grains, is just very similar to its ancestor, but it's sort of reduced a little bit. We still have these prothallial cells. These are just sort of cells that are there, honestly. They're not that functional or anything, but they're sort of these pancake thin cells. And then we have a generative cell, and then we have a tube cell, and that's literally it. Those are the only cells inside of a pollen grain. The tube cell is sort of this structure that's quite large, actually, here. And it's got a nucleus, and then the generative cell is in black here, and it also has a nucleus, but it sort of almost looks like it's literally inside of that tube cell. Well, it kind of is, actually. So anyway, over evolutionary time, there was a reduction of the microgametophyte. That's the main point of this. So let's look at where those pollen grains come from. This is a pollen cone. This is a longitudinal section through one of those small pollen cones that I showed you pictures of on the previous lecture video. And there we go. Another word for a pine cone is actually a microstrobolus, right? This whole thing is a strobolus. It just really kind of means cone, and it's a microstrobolus because it produces microspores, and it's got microsporophylls. Remember this sort of same exact terms from the Selaginella strobolus. We've got a microsporophyll, which is really just a leaf. And then we also have a pollen sac here, which is the microsporangium. Right, so that's not new. The microsporangium attached to a microsporophyll that's producing microspores, that's not new. That's we learned last week. Now we can just apply these other terms, these sort of newer terms, the pollen sac and the pollen grains. So a pollen sac is a microsporangium and a pollen grain is a microspore. Cool. So now looking at sort of zooming in on this smaller part of the life cycle here. We have meiosis happening in a microspore mother cell, and meiosis occurs and it produces 4 of these microspores. Each one of these 4 microspores will become a pollen grain. Cool. And we're going to get to this lower part here in a minute. Actually, no, I'll say this right now. So similarly, on the female side of the story, we have, again, 4 spores, 4 megaspores this time instead of microspores, but only one of them survives, so we only have one functional megaspore, unlike in the pollen grains. Okay, and then the other big difference between the male and female structures here is that the megaspore is attached to this really usually quite heavy-duty ovuliferous scale, which itself has this bract. So more on that in a little bit. So more on this pollen anatomy. We've got pollen grain. It's literally just another word for pollen, or for microspore, and it has a highly reduced microgametophyte inside of it. The microgametophyte is just this. It's literally just these 4 or 5 cells. The prothalial cells, the generative cell, and the tube cell. That's it. Yeah, so there's 2 or 3 prothalial cells. Now, conifers, a lot of conifers, including the pines, sort of have these really convenient big air bladder sacs attached to this microspore. And this is nice because it can help them catch the wind. Of course, this is what they need to do now that they are relying on the air to move sperm cells around. In other words, pollination. So then we have one more mitotic division in the microgametophyte. The generative cell divides and it produces 2 sperm cells. Okay, so now, pollination. Talked about this last video, but let's talk about it again. Pollen is produced inside of pollen sacs and contains microgametophytes. These microgametophytes are transferred to the ovule and enter the micropyle, reaching the nucellus, okay? So once that pollen grain has reached the nucellus inside of the micropyle and pollination has been completed, then fertilization can start, okay? So pollination and fertilization are not the same thing. Pollination happens, this is the act of getting a pollen grain to an ovule. But fertilization is the act of a sperm cell and an egg cell uniting. So for fertilization, the microgametophyte contained inside grows a pollen tube by elongation of the tube cell through the microspore wall. So now it will bust through that microspore wall. The pollen tube will grow and germinate. Remember, this is all happening literally while this pollen grain is sitting in the micropyle. In other words, right here. Remember the micropyle, sort of that little opening in the integuments. This tube starts to form only once it has come into contact with this ovule. So once there, the tube cell nucleus will direct the growth of the pollen tube, and then the 2 sperm cells will follow it down that tube. And then there you go. The sperm cell can sort of fuse with the egg cell, which is inside of the archegonium. Okay, so now, let's just say that happened to this one that turned green right here. Nice. This is our zygote. This is our first diploid cell in a while, actually. It's all the way since that Megasporangium, I think. So the zygote here, just one of the eggs was fertilized. The other one was not. Well, over time, there's going to be some mitotic divisions of that one zygotic cell, and it's going to form an embryo, and that's what we see here. This on the right is an older seed. So on the outside of the embryo, seen in green, we have the megagametophyte, okay? And then the outside of the megagametophyte is the seed coat. The seed coat has multiple layers, including that very outer integument that we talked about a long time ago, the megasporangium, and also, I guess, in theory, that megaspore membrane, which is possibly very thin. Okay, the embryo, the megagametophyte, and the seed coat together, these make a seed. This is a seed, finally. Yay. Okay, so this is what's so awesome about seeds. It's literally 3 stages of the life cycle all in one, right? The very oldest part here is that seed coat, and it's from the original sporophyte plant, right? This is all happening on a strobolus. And then the gametophyte germinates literally right inside of that megasporangium, as we talked about. And then after fertilization takes place, the next generation is that embryo. And so we actually have sort of 3 phases of the life cycle in one. And now we sort of have this embryo that's living inside of its mother, if you will. It's living attached to its mother plant. And so it's able to get nutrients maybe much easier than some other embryo of some seedless vascular plant that only had its little gametophyte that was very dependent on the environment. Now we've got all these extra layers. The integument can be very hardy and can allow for the, very hot or cold temperatures or dryness or long periods of time. Very cool. So we do have a couple of new terms. These should be added to your list of terms from the beginning. We've got cotyledons. Here's this new term, cotyledon. It literally just means an embryonic leaf. So it's the very first leaves that form. Maybe there's 1, maybe there's 2, maybe there's 5 or 10. The cotyledon or cotyledons are those embryonic leaves. Okay, and then we have terms, remember? The ram and the sam, the root apical meristem and the shoot apical meristem. Yeah, those are the first things to happen sort of in an embryo. The first differentiation of cells will be sort of the top and the bottom. And in the middle of the sam and the ram, we have this sort of other structure. It's really not technically the root or a stem, and it's called a hypocotyl. Okay, this is sort of where everything's got to sort of attach, all of the above ground stuff and the below ground stuff, if you will. But the hypocotyl is probably it's not really noticeable very often once the sporophyte is starting to grow. Oh yeah, and another word for the shoot apical meristem inside of a seed is an epicotyl. The other word for the root apical meristem inside of a seed is a radicle. All right. So these are all the parts of a seed plant embryo. All right. And again, a little review. Okay, so now let's sort of bring all of this together. Kind of bringing together the last lecture video and everything I've been talking about so far on this video. This is the old life cycle of a seed-free vascular plant of Selaginella, the other heterosporous, sort of that other seed-free heterosporous diplohaplontic plant. Remember, with them, the megaspore is released from the megasporangium and it sort of lands on the ground, and it's the megagametophyte will then therefore germinate sort of on the ground wherever the megaspore landed. And then again, the sporophyte, right? So the actual, the thing that we think of as the plant, like the bigger photosynthetic vegetative structure, is relatively limited to being grown right where that megagametophyte was growing. The gymnosperms kind of figured out a way to deal with that by retaining the megagametophyte in the megasporangium and therefore on the megastrobolus on that cone. So therefore attached to the tree itself, right? It's all, the seed is forming on the mother tree, if you will. Cool. All right. So we've talked about a lot of the parts of this life cycle sort of independently. What else do we need to talk about? Oh, here's a review slide for you to review on your own. And this is everything we've been talking about, but it's sort of in that other form to have it in I wanted you to be able to contrast this with the other life cycles we talked about with Lycopodium and Selaginella. And whatever else I did with this. Oh, the bryophytes. Okay, so we have two very different types of cones here. We have male cones and female cones. In this picture, at least, the male cones are these numerous tiny structures here, okay? And those are also known as microstrobili or microsporangiate strobili, or pollen cones. Okay. And then we actually have two very different stages of the ovulate cones in this picture right here. We have the very, very young ovulate cones right up there. Those are probably in the pollination stage. Very young, very small. And by next year, they're going to look like this. There's a year or probably a year old ovulate cone. Cool. Okay, so here's what a section of an ovulate cone would look like. You've got all these repeating units. So a just like a pollen cone was a microstrobolus, a ovulate cone is a megastrobolus. And another word for that is a megasporangiate strobolus. Again, we've got that cone axis, and yeah, notice how similar these cones look when we look at a cross-section. We've got these overlapping units. Okay, so how does this whole cone connect with the whole ovule story that we were just talking about? We're talking about two very different scales of things here on this video. Well, this is a mature ovule in this picture, and a mature ovule is situated at the base of an ovuliferous scale. Okay, and an ovuliferous scale also sort of has this sterile bract associated with it, and then this unit of these structures right here is repeated over and over again on an ovulate cone. And that's what we're seeing here. So really there'd be sort of an ovule right here, ovule right here, ovule right here. Okay. Or maybe a pair of ovules or something, depending on the species. Now this is a very a two-dimensional look at something that's very three-dimensional, right? When we think of a cone, I happen to have one right here. Yep. When we think of a cone, right, it's something that's very, very three-dimensional. But in this picture, I have sort of imagined, I've just taken like a very nice sharp saw and I've sliced right through it. And so we're able to pick apart these individual units. And really, if I were to rip one apart, I'm not going to do that because this is one of my special cones. But if I were to rip this off right now, this ovuliferous cone scale right here, then I'd find either some seeds or more likely some seed scars where there at one time were seeds. Okay, now here's another kind of crazy thing. This whole repeated units thing of an ovuliferous scale with an ovule and a subtending bract, this is actually literally evolutionarily, it is a branch. It's literally, these are small branches that are that are repeated over and over again, and they're highly, highly modified, so they don't look anything like actual branches. But if we were to look at sort of their evolutionary history and, you know, how these things were formed, well, remember the sporophylls, those were really just leaves in the seed-free vascular plants. And really what happened was those leaves have axillary buds and they branch, and that's sort of what an ovuliferous scale is. And then the ovules just are part of that story. Okay, right. So it's really a modified shoot with the leaves and stems reduced and fused together. That's what this whole unit is. This whole one unit right here. And that means a cone, a strobolus, a megastrobolus. It's just a branch with a bunch of branches. Okay. Hopefully that wasn't too mind-blowing. Okay, a bract is just a leaf that subtends a reproductive structure. Okay. So we've got development from a young ovule to a mature ovule. And just a quick review, the megasporocyte is that very first, that very, very rather last diploid cell. This is the cell that's going to undergo meiosis. Right? And then it's going to form one single functional megaspore because 3 were aborted. And then it's the mitosis of that one functional megaspore that's going to result in a megagametophyte. And then mitosis of that megagametophyte is going to result in an archegonium. Mitosis of the archegonium is going to result in a cell, in an egg cell. Cool. So the pollination stage in pines is when sort of before actually meiosis even happens, or maybe at the very early stages of meiosis from the female side, from the female's perspective. So pollination is happening when there's still a megaspore mother cell or a megasporocyte. This stage, this would be a picture, an image of one of these ovuliferous cone scales in an ovule, sliced very thinly with a nice razor and dyed nice color so that we can really see the individual cells that make this up. Okay, and then if we sort of look at the diagram here, the main kind of chunk hanging off here is that scale. And then we've got the whole ovule that includes the integuments, the megasporangium, and at this stage that megaspore mother cell. What does M stand for here? Yes, that is the micropyle, that little opening that needs to be present, because during pollination, a pollen grain is going to be flying through the air and it's going to land right by that micropyle until it's sort of taken in to the ovule and pollination is completed. And at that stage, the micropyle will sort of close off with the pollen grain inside of it so that there's no other sort of competition. So this is right after pollination and probably right before fertilization. Or, well, not right before fertilization, but right before the megasporocyte is probably going to undergo meiosis. Yeah, so the whole time that pollen grain is just sitting there while the ovule is developing, like for a year or maybe less than a year. It really depends on the species, but probably up to multiple years for some species. The pollen grain is just chilling. It's just waiting for its moment to germinate that pollen tube and then to have its sperm cells kind of travel down that pollen tube and find an egg. Yeah, they have to wait for the young ovule to develop into a mature ovule. That whole process that we just talked about. Okay. So pollination versus fertilization. Fertilization is the same thing we've been talking about. It's when a sperm cell and an egg cell meet. Okay. So this is sort of what fertilization looks like in a real, again, like sort of this is a picture through a microscope, a perfect timing, a perfect sort of slice of time through when a pollen tube is growing. And then actually we have here the sperm nucleus coming into contact with the egg, or it's about to come into contact with the egg. So fertilization is about to happen in this picture. Very cool. And then, of course, that once fertilization does happen, a zygote will form, and then it's going to form an embryo, right? The zygote's going to form an embryo, and then an embryo inside of a megagametophyte, inside of a seed coat, is, of course, a seed. Okay, so here's just another sort of review slide talking about some of those terms from earlier. And I think that's my last slide. It is. So thank you for your attention on this long video of seeds. And please don't hesitate to stop by my office hours this week if you wanted to chat about anything related to gymnosperms or seeds. Hope to see you there.